Breast cancer remains one of the most formidable adversaries in modern medicine, a disease defined by the uncontrolled growth of cells within the breast tissue. It is not a single entity, but a complex collection of diseases, each with its own genetic signature and behavioral pattern. For decades, the fight against this malady has been one of relentless scientific pursuit, leading to therapies that can target the very mechanisms that allow these cells to thrive. Yet, even as our understanding deepens and our treatments become more precise, a persistent and often tragic problem remains: drug resistance. A patient may initially respond with remarkable success, only for the tumor to defy the treatment and return, often more aggressively than before. This phenomenon of resistance is a testament to the cunning adaptability of cancer itself. The tumor is not merely a static mass of malignant cells, it is a dynamic and evolving ecosystem. Within this environment, cancer cells can mutate, adapt, and develop new strategies to survive the chemical onslaught of therapy. This evolutionary arms race is one of the greatest challenges in oncology. Understanding why a drug that once worked so well suddenly fails is a central question that preoccupies researchers and clinicians alike. The answer, it seems, may not lie solely within the cancer cells themselves, but in the complex world they inhabit. A world that we are only now beginning to explore with the necessary tools and perspective. The search for answers has pushed scientists to look beyond the human genome and consider the vast, unseen world of microorganisms that live within us. For a long time, tumors were thought to be sterile environments, devoid of the bacteria, fungi, and viruses that colonize other parts of our bodies. This assumption, however, is being rapidly overturned. We are discovering that tumors, much like our gut or skin, have their own unique microbiome. This realization opens up a startling possibility. What if these microscopic tenants are not just passive bystanders? What if they are actively participating in the life of the tumor, influencing its growth, its interaction with the immune system, and most critically, its response to our most potent drugs? This paradigm shift forces us to reconsider the very nature of cancer. It suggests that a tumor is a composite organism, a collaboration between malignant human cells and a community of microbes. The implications of this are profound, suggesting that the story of drug resistance is more intricate than we ever imagined. It is a narrative not just of cancer cell genetics but of a complex interplay between host and microbe. The battle against cancer, therefore, may require us to fight on a new front, targeting not only the cancer but also its microscopic allies. This emerging field of research is beginning to illuminate the dark corners of therapeutic failure, offering new hope where it is desperately needed. Among the various subtypes of breast cancer, one of the most studied is characterized by an overabundance of a protein called human epidermal growth factor receptor 2, or HER2. In a normal cell, HER2 receptors sit on the cell surface and play a role in signaling pathways that control growth and division. However, in about 15 to 20% of breast cancers, a genetic error leads to the creation of far too many of these receptors. This HER2 positive status transforms the cancer cells, making them more aggressive, faster growing, and more likely to spread to other parts of the body. For years, a HER2 positive diagnosis was associated with a particularly poor prognosis, a dark mark against a patient's chances of survival. The discovery of the HER2 protein's role was a watershed moment, as it provided a clear, identifiable target. This led to the development of a revolutionary drug called Trastuzumab, a monoclonal antibody designed with exquisite precision to find and block the HER2 receptor. When Trastuzumab binds to HER2, it acts like a key breaking off in a lock, preventing the receptor from sending its relentless grow and divide signals into the cell's interior. For many patients, the introduction of Trastuzumab was nothing short of miraculous, turning a highly aggressive cancer into a manageable and sometimes curable condition. It became a cornerstone of treatment for HER2-positive breast cancer and a shining example of the power of targeted therapy. However, the triumph of Trastuzumab was not absolute. A significant number of patients, up to 35%, show little to no response to the drug from the very beginning. This is known as primary resistance. Even more troubling is the phenomenon of acquired resistance, where a tumor that initially shrinks under treatment eventually learns to overcome the drug's effects. For patients who do respond, a staggering 70% may see their cancer develop resistance within a year. The tumor, in essence, finds a way to hotwire its own growth signals, bypassing the roadblock that Trastuzumab has created. The reasons for this failure have been a source of intense scientific investigation, with most theories focusing on genetic mutations within the cancer cell itself. Strikingly, recent research suggests that it was hiding in plain sight, residing not only in the cancer's DNA, but also within the microscopic life forms that have taken up residence inside the tumor itself, silently contributing to its defiance. The idea that bacteria could live deep within a tumor was, until recently, considered highly unlikely, if not impossible. The prevailing wisdom held that such environments were sterile, shielded from the microbial world. However, with the advent of more sensitive genetic sequencing and imaging technologies, this dogma has been shattered. Scientists began to find compelling evidence of bacterial DNA within tumor samples from various types of cancer, including breast cancer. What was first dismissed as contamination from the skin during surgery or from laboratory equipment was proven, through painstaking research, to be a genuine biological phenomenon. These were not just transient visitors, they were intratumoral bacteria, microbes living within the cancer cells themselves. Among the various bacterial species identified, one has emerged as a particularly frequent resident of breast tumors, Pseudomonas aeruginosa. This is a common bacterium, known for its ability to thrive in diverse environments and for its role in hospital-acquired infections. Its presence inside a breast tumor was puzzling. What was it doing there? Was it merely an innocent bystander, or was it playing an active role in the tumor's biology? Researchers at the Chinese Academy of Science, led by Xiaohong Fang, decided to investigate this question, focusing on the possibility that this bacterium could be communicating with the cancer cells and influencing their behavior in ways that could explain the stubborn problem of drug resistance. Their investigation focused not on the bacterium itself, but on the chemical signals it produces. Bacteria, much like our own cells, use molecules to communicate with one another in a process called quorum sensing. One such signaling molecule produced by Pseudomonas aeruginosa is a small fatty acid derivative known as N,3-oxododecanoyl homoserine lactone, or 3-OC for short. This molecule helps the bacteria coordinate group behaviors. The researchers hypothesized that this bacterial signal might not be confined to an internal bacterial conversation. They suspected it might be overheard by the surrounding cancer cells, potentially altering their fundamental biology and, crucially, their vulnerability to drugs. To test this, scientists needed to confirm that both the bacteria and their signaling molecules were physically present in human tumors. Using sophisticated techniques like quantitative polymerase chain reaction, or qPCR, and fluorescence in situ hybridization, or FISH, they carefully analyzed tissue samples from breast cancer patients. Their findings were definitive. They detected Pseudomonas aeruginosa in 7 out of 34 tumors. Even more directly, using a highly sensitive method called mass spectrometry, they found the 3-OC molecule itself in 8 out of 24 tumor samples. The evidence was mounting. A known bacterium and its specific chemical messenger were indeed present in the very environment where cancer drugs were failing, setting the stage for a groundbreaking discovery. With the presence of and its signal, 3-OC confirmed in breast tumors, the next logical step was to determine what effect, if any, this molecule had on cancer cells, particularly in the context of Trastuzumab therapy. The research team conducted a series of elegant experiments in the laboratory using breast cancer cell lines that were known to be sensitive to the drug. They exposed these cells to the 3-OC molecule and then treated them with Trastuzumab. The results were striking and, honestly, deeply concerning. The presence of the bacterial signal molecule made the cancer cells significantly more resistant to the drug. Even cells that were once easily killed by Trastuzumab now survived and continued to thrive. What was fascinating about this process was that the 3-OC molecule was not toxic to the cancer cells on its own. Unlike a poison, it did not kill them or even slow their growth directly. Instead, it acted as a subtle influencer, fundamentally changing how the cancer cells responded to the therapeutic attack. It was as if the bacterial signal was whispering a secret to the cancer cells, teaching them a new survival trick. This discovery pointed to a novel mechanism of drug resistance, one that was not driven by a genetic mutation in the cancer cell, but by an external signal originating from a non-human microbial source. The bacterium was acting as a hidden accomplice, providing the tumor with a shield against our best medicine. To understand how this was happening, the scientists delved into the genetic machinery of the cancer cells. They analyzed which genes were turned on or off in the presence of 3OC and found a clear pattern. The bacterial molecule was activating two critical signaling pathways within the cancer cells, the TGF-β pathway, and, consequentially, the very same RBB2 pathway that Trastuzumab is designed to shut down. In essence, while Trastuzumab was blocking the HR2 receptor at the front door, the 3OC molecule was sneaking in through a back window, using the TGF-β pathway to reactivate the RBB2 signaling cascade from downstream. The cancer cell's growth engine, which should have been silenced, was being switched back on by a bacterial command. This mechanism provided a plausible explanation for how tumors could become resistant. The presence of could effectively render Trastuzumab useless. To confirm this, the researchers performed another crucial experiment. They used a second drug, this time an inhibitor designed to block the TGF-β pathway. When they treated the cancer cells with both the TGF-β inhibitor and 3OC, the resistance vanished. Trastuzumab was effective once again. This demonstrated a clear chain of command. The bacterium releases 3OC. 3OC activates TGF-β in the cancer cell. TGF-β reactivates RBB2 signaling, and the tumor becomes resistant. Breaking any link in that chain could potentially restore the drug's power. The discovery that a common bacterium can chemically instruct a breast tumor to resist a life-saving drug opens an entirely new frontier in the war against cancer. It confirms that a tumor is not an isolated island of malignant cells, but a complex ecosystem where human and microbial cells coexist and communicate. This research provides a tangible mechanism for a phenomenon that has long puzzled clinicians, and suggests that the tumor microbiome is not just a curiosity, but a clinically significant factor in treatment outcomes. The finding that tumors from patients who responded poorly to Trastuzumab were more likely to contain provides a powerful link between the laboratory finding and the real-world challenges faced in the clinic. The implications of this work are far-reaching and honestly offer a profound sense of hope. If bacteria can contribute to drug resistance, then targeting these bacteria could become a powerful new strategy to improve cancer therapy. It suggests a future where treatment plans are far more holistic. A patient with HER2-positive breast cancer might not only receive Trastuzumab, but could also be tested for the presence of intratumoral. If the bacterium is found, treatment could be supplemented with antibiotics or with drugs that specifically block the 3OC signal or the TGF-β pathway it activates. This approach could disarm the tumor's microbial accomplice, making the cancer vulnerable to therapy once more and potentially preventing or reversing resistance. This research also prompts a cascade of new questions that will drive the field forward for years to come. Does the 3OC molecule affect resistance to other cancer drugs or in other types of cancer? Are there other bacterial species producing other signaling molecules that have similar or perhaps entirely different effects on tumor biology? Understanding the full scope of the tumor microbiome's influence will be a monumental task, but it is a necessary one. Each discovery brings us closer to a more complete picture of cancer, not as a disease of our cells alone, but as a complex interplay of genetics, environment, and the trillions of microbes that share our bodies. Ultimately, this breakthrough is a powerful reminder that in science, the most profound answers often come from looking where no one thought to look before. By listening to the subtle chemical whispers between bacteria and cancer cells, we have uncovered a new dimension of drug resistance. This knowledge does more than just explain a problem. It illuminates a path toward a solution. It fosters a future where we can fight cancer on multiple fronts, extending the effectiveness of our best therapies and offering renewed hope to patients and their families. The unseen world within the tumor is no longer silent, and by learning its language, we may finally learn how to win the fight. Thank you for watching. Subscribe for more breakthroughs in biology.